One stop. One stop science shop. Hi, I'm Mercy, and welcome to the one place where we try and answer all your science-related questions. It's the one-stop science shop. Let's see who has a question for us today. It's Tom from Vietnam. She asks, how long is our DNA? That's an interesting one, Tom. Don't go anywhere while we at the One-Stop Science Shop try to find you an answer. Our DNA can stretch from the Earth to the Sun 600 times. This is because there are 100 trillion cells in the human body and each DNA strand is 6 feet long. So imagine what would happen if you unwound and linked them all together. DNA makes RNA and RNA makes proteins. This is the basic principle on which our bodies function. And that is how genetic information flows in human cells. I guess most of us have heard of DNA. That's the part of the cell which contains all the genetic information. Even the strawberry contains DNA. You don't believe me? Check this out. For this experiment, this is what you need. Some water, a cheesecloth, clear shampoo, common salt, a Ziploc bag, measuring cups, empty glasses, rubbing alcohol, and strawberries. First of all, we're gonna make the extraction solution. For that, you'll need 450 ml of water, which I've already measured out here, 50 ml of clear shampoo, which I'm gonna add to the water. There you go. Be careful not to spill any. And a teaspoon of common salt. Stir it gently to prevent frothy bubbles forming. There we have our extraction solution. Now I'm gonna take the Ziploc bag and put 5 strawberries and 5 and put them in the Ziploc bag. We'll measure 100 ml of our extraction solution. And add it into the Ziploc bag. Now let out the air and lock the bag properly. And now comes the fun bit. Just squish the strawberries gently. All the squishy strawberries. I feel like eating one. So you might have to do this for a good 5 minutes till you get all the strawberries juicy and with the right consistency like this, looking like this. But be very gentle because it might burst. Now we're gonna play detective. We're gonna use the cheesecloth, put it on top of the empty glass, and use a rubber band to tie it up. And very, very carefully pour out the solution. This can get messy, so be very careful. Squeeze out as many juice as you can. Now we'll take 25 ml of rubbing alcohol or isopropyl alcohol. And gently pour it into the glass. Let's see what's happening here. I can see the DNA strands. The DNA is collected between the layer of alcohol on top and the strawberry extract underneath. The DNA is insoluble in alcohol, so it precipitates. What you see here is the precipitation of strawberry DNA, long thread-like DNA molecules at the interface of the alcohol and the DNA solution. You should try this at home. Here's what you need. A Ziploc bag, a cheesecloth, clear shampoo, common salt, empty glass, measuring cups, water, rubbing alcohol, and strawberries. Here's what we learned from the strawberry experiment. It has DNA like every living creature. Hey, looks like we have a visitor.
Oh wow, this place is amazing.
Hi, I'm Mercy. Come on in. Welcome to the one-stop science shop.
This place is nice.
What is your name and how old are you?
My name is Awang and I'm 7 years old.
And what class are you in?
1B.
So what brings you here to the one-stop science shop today?
I've heard that ulcers help the curious and I'm curious to find out much more about RNS.
Oh my god, that's excellent. I'm gonna introduce you to some woman who is very special and she's a pioneer in this field.
She works in the field?
No, actually she works in the lab. Let's see what we got. This is Dr. Narry Kim, a South Korean biochemist and microbiologist, best known for her work and understanding of microRNAs, which play key roles in the regulation of gene expression. But what exactly is RNA? Hang in there and all will be revealed. She was born in Seoul, Korea in 1969. Her father and teachers wanted her to become a doctor, but instead she decided to study biology. Kim first became interested in science as a high school student and went on to study in Seoul, Oxford, and Philadelphia. In 2002, her research group uncovered the formation process of microRNAs in cells. So what are RNAs? RNA stands for ribonucleic acid. RNAs enable a cell to use the instructions in the DNA to make enzymes. It is chemically more reactive than DNA, which makes it the more suitable molecule to take part in cell reactions. Kim's pioneering studies have helped in the understanding of microRNAs better and have discovered that they are involved in nearly all the developmental and pathological processes in humans and animals. Unfortunately, when they don't work properly, they can cause many diseases. In 2013, Dr. Kim, along with Professor Jin Soo, helped develop new technology to eliminate specific microRNAs. This has the potential to be used to try and cure illnesses such as cancer. In the research, special enzymes were used to separate certain microRNAs from a cell. When this was applied to cancer cells, the proliferation rate dropped to 1/3. Let's hope that the research continues to be successful. Dr. Kim has been recognized for her work with national awards from the South Korean government and a Women in Science award from UNESCO. She currently heads her own RNA biology lab in Seoul and hopes that her achievements will inspire many more young Asian women to become scientists. When she got the phone call giving her the news that she had been elected for the Women in Science award, she was in her pajamas, pillow fighting with her kids. It must have been the most unexpected phone call of her life.
That was really interesting. Not a field inside. She's not only a pioneer in RNS research, but also in helping more women scientists.
Maybe you want to follow her footsteps someday?
Who knows? Let me start by telling you a fact about RNS. About 5% of the weight of a human cell is RNA, whereas only about 1% of a cell consists of DNA.
Thank you for telling me that. Now, for all the budding researchers out there, you need a very special tool if you intend to study macromolecules like RNA.
A microscope.
Do you want to build one with me right now?
Now?
Yes, no time like the present. Let's see what we have in our one-stop science box today. Hey, here we go.
A laser light.
Yeah, let me put it there for you. That's a white tray.
A white tray. A jar of water.
Jar of water.
What's this?
It's a glass tumbler.
A glass.
It's a stirrer.
Stir. A spoon.
Thank you. A spoon. Clothes pegs.
Okay. One more.
All right, thank you very much.
And a glass.
Thank you, Nawang. Okay, let me get this out of the way.
How on earth can this make a microscope?
Well, you just have to take this thing out and I'm gonna show you how. Okay. First of all, we're gonna take a glass of tap water. And then Nawang, would you like to remove 3 pegs because we need only 1?
Okay, 1, 2, And 3.
Thank you very much.
Put this in the side.
Alright. So what we're gonna do is we're gonna dip the stirrer in the water and stick it with a peg. Make sure there's a drop of water at the end of the stirrer, okay? And then, do you want to help me shine the laser light at the droplet? Nawang?
Yes?
Now look at the water shadows there.
Oh, this is unbelievable. The water looks clean, but it's a little bit dirty.
But the water is not really dirty, but there are lots of microorganisms inside it.
Oh, that was fantastic. How does the laser magnify the organisms?
The laser is already very concentrated light, with the light around it giving it the shape.
But what color laser should we use?
Try using a darker color, as this will make the negative images easier to see.
That was fascinating.
I'm glad that you enjoyed it. Who knows, you might be a microbiologist someday.
I am going to seriously think about it.
That's wonderful. And if you want to try this at home, here's what you need. A jar of water, white tray, clothes pegs, glass tumbler, stirrer, a spoon, a laser light, glass. I think we really got down to the core of molecular biology today. Thank you for being with us. See you next time on the One Stop Science Shop.